Hello guys. Today we are going to discuss about a laboratory test called CALR gene mutation exon 9. Why do we do this test? To help diagnose bone marrow disorders known as myeloproliferative neoplasms, in which the bone marrow produces too many of one or more types of a blood cells. Calreticulin gene, called CALR for short, is responsible for making a protein called calreticulin. The exact function of a calreticulin protein remains largely unknown, but it is likely involved in ensuring the correct folding of a new protein, maintaining correct calcium level in cells, and a number of other cells function. Mutations of CALR gene are associated with bone marrow neoplasm that causes the production of too many blood cells. These blood disorders are collectively known as myeloproliferative neoplasms. The CALR mutation test looks for abnormalities in CALR gene to help diagnose and classify MPN. The MPN most commonly associated with CALR mutations are essential thrombocythemia or ET. There are too many platelet producing cells, megakaryocytes, in the bone marrow. Primary myelofibrosis, also known as chronic idiopathic myelofibrosis. There are too many platelet producing cells and cells that produce scar tissue in the bone marrow. CALR mutations are the second most common genetic abnormality after JAK2 mutation associated with essential thrombocythemia or primary myelofibrosis. They are present in about 20 to 25% of the adult with ET and 25 to 30% of adult with PMF. Although rare and not well understood in children, 50% of pediatric PMF patients had CALR mutations. The CALR mutation is acquired after birth as opposed to inherited. It is caused by the addition or removal of a small amount of a genetic material to a region of a gene called exon 9. This lead to an abnormal calreticulin protein. It is not yet understood how the mutation or mutant protein lead to signs and symptoms of MPN. While more than 50 types of CALR mutations have been found, only two most common variants associated with MPN are included in conventional PCR testing. Next generation sequencing based analysis should detect all mutations variant but is not widely available. In addition to helping diagnosis of MPN, CALR mutation testing can provide information about a person's prognosis. Studies have shown that compared to individuals with JAK2 mutation, those with the CALR mutation had a milder disease course, fewer signs and symptoms of a blood clotting, and better survival. Sample: a blood sample taken from a vein in your arm or sometimes a sample of bone marrow is required. Normally, it is not required for healthy persons. That is about CALR mutation.